Hi everyone, I hope to be finishing up the last lecture for chapter 2. So we're going to be discussing the experimental variables, which include our independent variable and our dependent variable. Remember, a variable is something that changes between participants. Remember that an experiment is a way to test cause and effect relationships. So the independent variable is the variable I'm assuming to cause the change, and the dependent variable is the effect variable. So this is what causes the change, and this is the effect of the change. This is what we call manipulated by the researcher. I control who gets it, how much, when they get it. The dependent variable is what we call a measured variable. So if I'm looking to study the effects of television violence on aggression, then my causal variable is the television violence. My aggression is my dependent variable. So the television violence is my independent variable, and my manipulated variable, I decide who's in the control group, who's in the experimental group, what the control group watches, and what the experimental group watches. And then the aggression is my dependent variable, and I measure aggression in both groups. So if I'm looking at the effect of a drug on depression, then I manipulate who gets the drug, who doesn't get the drug, how much of the drug they get, when they get it, where they get it, and how they get it. So I'm manipulating that variable. My control group gets a placebo, my experimental group gets the drug, and I assume the drug causes a change in depression levels. And I measure depression in both groups, compare the groups, and see if my drug group has lower depression than my control group. There are also extraneous variables in studies, just like we talked about third variables and correlations that can kind of confound the results and you can't clearly see why that relationship exists. Extraneous variables are third variables that I'm not controlling. I'm not interested in studying, but I want to hold them constant so they don't systematically influence my results. Because then I don't know that my IV caused the change in my DV. It could be some other variable that I didn't account for. So really important to really carefully think about and control those extraneous variables. For instance, if I am doing an investigation of television violence on aggression, I might want to hold children out or restrict my study so that children with behavioral issues and challenges are not part of my study because they may automatically be a little more aggressive, something of that nature. Or a child that was subjected to trauma or child abuse at home or something of that nature, I might want to exclude them from my study. So with regard to the whole process, I have a hypothesis that more violent television is going to influence the aggression levels. I randomly assign my participants to either the control or experimental group. My control group watches non-violent shows. My experimental group watches violent shows. I then expose them to a situation where I can measure their aggression levels, maybe with a toy, maybe with peers on the playground. In this case, I'm going to put them in a room and see how they interact with the doll. And I'm going to see if the control group beats up the doll as much as the experimental group. If the experimental group is much more aggressive after the study, then I can conclude that my independent variable or my violence within the television shows is what caused the higher aggression levels. So again, you have a population. You randomly select a sample that should be representative. I'm going to select a sample of 100 people. I'm going to randomly assign them to either the control group or the experimental group. 50 people are in each group. I'm going to expose the control group to non-violent television or a placebo. I'm going to expose the experimental group to the independent variable or the drug, or in this case, violent television. And then I'm going to compare both the experimental and control group on the dependent variable. So I'm going to measure their aggression levels in both groups and see if one group is higher. If in fact the experimental group is higher in aggression than the control group, it's said that my experimental group was influenced by my treatment or my independent variable, which in this case is the more violent television show. So the reason that carefully controlled procedure and that random assignment and that random selection, they all are what's so powerful and allows you to say that your IV caused a change in your DV. But there are some challenges with experimental research. As I mentioned, we do have experimental bias, so we try to do blind studies where the researcher does not know who's in which group, or double-blind studies, blind studies where the participants don't know, and double-blind studies where the participants or the researchers don't know. Or I might have blind observers, so I might actually have observers coding the aggression levels of the children, and they don't know which children saw the violent shows and which children saw the non-violent shows. I also want to make sure I'm using ethnocentric researchers, and I have research teams that include people from different cultures so that we're not interpreting it from our biased perspective. I also want to make sure my sample is cross-culturally representative so that I can make sure I can generalize to a larger population. For my participant, again, that sample can be biased if I'm only using men or I'm only using smart people. So I want to do random sampling where everybody has a chance to participate in my study, and whoever participates is purely random. I'm not influencing them one way or another, and it's representative and such that it represents all the different diverse variables: gender, age, ethnicity. And then once I have that sample, I randomly assign them to control or experimental. And then for participant bias, I want to make sure they're giving me authentic responses, they're being genuine, so I maintain confidentiality, and then I let them complete their surveys anonymously, so I don't have a name attached to their data, and then that helps them have some protection so they can be honest and forthright. And then additionally, I use double or blind studies, and then I use placebos as well. When we have an experimental finding, they're submitted to, correlations are submitted to that Pearson product moment in most circumstances, and you get that R value, that correlation coefficient. For experiments, it really depends on how many variables you have, how many groups you have, because you can have more than one experimental group. And so you use the appropriate statistical procedure for that research design. And the statistics tell you whether or not there's significant differences in the group or there's meaningful differences in the group. If they're not statistically significant, then they're not said to be meaningful. Maybe I weigh 1 pound more than you, but is that really meaningful? No. If I weigh 10 pounds more than you and we're the same height, that might be meaningful. So differences are considered meaningful if there's a less than 5% chance of getting that result statistically. So we use probability. And greater than 5% probability means it's just kind of normal. It's not really meaningful differences, so the results are not significant. Less than 5% chance means, wait a minute, those are rare results, and so they're meaningful and statistically significant. So statistically meaningful significant differences mean that my IV caused the differences, and so my treatment worked or didn't work. And any outside differences between the groups, we use random assignment to kind of disperse those evenly. We use that carefully controlled procedure, treat the groups equally, so that we know it's just our IV that's different between them, and therefore the IV should be the only difference. If there's a difference in the DV, it's purely because of that IV. So the differences in the dependent variable are said to be caused by the independent variable. Once I'm done with my research results, I can actually write up my paper consistent with APA guidelines. There is a manual that outlines all the rules for writing a paper and your formatting and your margins and how you reference a source citation and does this get capitalized, where do the commas go? And that's really important because there needs to be a consistency. If we all submitted them a million different ways, scientific journals wouldn't be able to easily sort them, compare them, and analyze them for publication. So we follow these guidelines. They're submitted to what's called peer-reviewed journal articles. They're academic journals. We do not use just like GQ magazine or Vogue magazine. They're peer reviewed, so they're academic journals, magazines essentially, and they publish purely scientific articles written by other psychological researchers, and it's really to keep us in the profession informed of current research. So they're written to an audience of scholars rather than to just kind of the general public. And what happens is you submit an article proposed for publication, your colleagues review it, they tend to have an expertise in the field as well. They look at kind of your methodology and your statistical analyses and your ethics, and they kind of scrutinize that. And they will sometimes submit recommendations for edits, and sometimes they'll just reject it altogether, or sometimes they'll approve it just the way that it is. So they analyze that work, the quality of the work, they give you feedback, and then the editor ultimately gets to decide whether they're going to publish the article and whether they're going to publish it with or without any revisions that were recommended. And this is really important because it allows replication. And remember, we said in the very beginning of this chapter that replication is critical to science, because we need to look at studies, look at their results, try to repeat those results. If we can't repeat the results, they don't have as much credibility. So experiments should be repeated multiple times under different samples, different conditions, and see if those results are consistent or reliable over time. And then the peer review process, make sure there's enough detail for replication to be possible. In the end, we're looking for reliability and validity. Reliability is we want the results consistent over time. It shows that the instruments or the tools are going to produce the same general scores over time. Validity is the tool is measuring what it's intended to measure. And so this is really critical that I'm ensuring a research method that's both valid and reliable. Finally, we're going to talk about ethics, and some foundational studies have really kind of inspired some ethical guidelines. The Tuskegee syphilis study, they actually injected syphilis into African Americans, and they let the disease take its course, and even long after we found treatment for it. Stanley Milgram's shock study, they had participants exerting shocks on other Confederates, and to the point where they thought they were giving him a heart attack. And it was there was deceit involved, and it made participants feel very guilty and bad. And then little Albert, they actually basically induced a phobia in him, and we'll talk about this in chapter 6, and we'll talk about this in the learning chapter, and we'll talk about this in the social psych chapter. Either way, APA establishes those ethical code of conduct that we have to adhere to, requires us to provide informed consent to all participants in a research study. In advance of taking or participating in our study, we have to tell them what's going to happen, what are the potential risks, dangers, how much time, and we have to give them the opportunity to withdraw at any time without any consequences. We're also required to provide them a debriefing at the end, where we again reiterate the risks to the study, where they can go for help if there's challenges. And if we used any deception, like if I'm going to do a memory study, I can't come right out and tell you I'm doing a memory study. So I may tell you I'm studying cognition, but then in the debriefing, I'm going to tell you specifically I was studying memory. So we tell you of any deception that we used, and we're much more clear and specific. We also need to adhere to special procedures for protected populations like prison populations, children, elderly, and animals. And all research has to be submitted to an institutional review board, which is a committee that we put a proposal to the committee. We say, this is the study I want to do. These are the tools I'm going to use. And they approve it and decide whether it's ethical or not and adds value. And then finally, we have to respect patient confidentiality in therapy. Everything you say is confidential unless you threaten yourself, another human being, you disclose that you're abusing a child, or you're abusing an elderly individual. So then we have to break that confidentiality. Animal research also has to have a special provision where they have an institutional animal care and use committee that oversees the treatment of the animals to ensure that it's humane at all times. And so that's kind of the end of chapter 2. I hope you have enjoyed learning about research. I hope you enjoy reviewing your own research study and the learning activity, and I'll look forward to answering any questions you have.